All right. So, uh, ladies and gentlemen, it's a pleasure to be your host in this morning or evening, depending where you are in the world. Um, my name is Diogo Berardo. I'm Director of R&D at NOVOS Labs. And this webinar is part of a webinar series, of which this one is the first webinar. And today we feature the Professor Satomi Miwa from Newcastle University. And our topic of discussion is going to be NOVOS Core in a rescue model of metabolic age. Next slide, please. First, a little bit of an overview with a little bit of disclosure. So the main topic of already in pre, and he has also passed peer review, so it should be published very soon. Is the main results are in animal model, and there is also in vitro supporting data. And of course, based on that, we cannot make claims on human clinical efficacy. Very quickly also, NOVOS Core is a multi-ingredient nutraceutical, and the nature of this talk is purely educational and doesn't constitute medical advice. Our particular conflicts of interest are disclosed in the manuscript itself, in the preprint that is already openly available. Next, please. So the learning objectives for this session. So today you should come out knowing this new rescue model of an obese, overweight, aged mice and why it's important for geroprotective interventions. You should also have a notion of the main findings regarding lifespan and health span after intervention with NOVOS Core, and also with the intervention of a senolytic combination. To know that, you also need to do the basics, which is what are senolytic interventions and what are xenomorphic interventions or xenostatic interventions. And then finally, the climax is that we do outline a translational hypothesis for combining NOVOS Core with the GLP-1 therapies in obesity care. Next. So we should take less than 50 minutes, and we are going to have a Q&A too. The sections of this presentation are going to be the following. So first, we're going to have a little bit of background and clinical framing. So we're going to talk about GLP-1s a little bit, just enough, and also very basic aging biology, so that we are all on the same page. Then I will pass the mic to Satomi, and she will present the study to us with a lot of results, including in-mice results and mechanistic results. And then finally, I will have the final word to talk about this translational potential that I alluded to. Next. So if you're an MD, why should you care? So this is preclinical data and in vitro data, so why should you care? Well, it's a new angle. So let's talk a little bit about obesity. So right now, obesity, we all know that it's not just about the weight gain. There is a lot of repercussions at the systemic level. Namely, the most widely known one is chronic inflammation, which eventually leads to an increase on the number of senescent tissues and cells, and also on the senescent signaling between these cells. And so even if one were to ameliorate or even cure the obesity epidemic, the repercussions of being obese early in life would still be present. And this is where we start from. So we think that we might have, is an hypothesis, we might have a way to address these repercussions of obesity. Next, please. So we cannot talk about obesity without talking about GLP-1 therapies. They started to be studied for type 2 diabetes, but then the second and the largest wave is, of course, in obesity. We all know the amazing trials that have been out with the amazing effect size in terms of weight loss. We also know that a lot of other things improve when someone actually loses weight. And we also know that there are some off-targets, beneficial off-targets of these therapies that, for example, the inflammation reduction cannot be fully attributed to the weight loss. And that's still not enough to address all the consequences of being obese, of ever being obese in life. Because, as I mentioned before, there's going to be accumulation of almost like this obesity scar, which is constituted by senescent tissues, and that can only be addressed from the paradigm of aging biology. So this is why we're interested in this, and this is our hypothesis that targeting aging biology pathways that are complementary to this GLP-1 mediated loss can potentially address this residual frailty biology and obesity. And why would that be so? So there are two lines of evidence that are quite interesting. One, as I mentioned before, is that GLP-1 therapies are having off-target beneficial effects in chronic diseases and age-related diseases. And why would that be? And the other one is that, as we'll see with this first study from Satomi, that aging therapies actually have a very positive effect in obesity models. And the reason seems to be that there is, of course, a relatively well-known overlap between the phenotypes of aging and obesity. So some parts are unique, some parts are in common, and our hypothesis is that we are targeting the common parts, especially if we combine both, for benefit of both conditions, both aging and obesity. So I've been talking about the biology of aging and aging, so I just want to give a little bit of background on it. Next, please. Aging is, the status quo is that we see it through this framework of the hallmarks of aging. So there are about 12 concepts that are all interrelated, that they can accelerate or delay the aging process. So NOVOS Core itself, which is one of the interventions that we're going to be discussed, was formulated using natural compounds to address as many of these hallmarks as possible. Next, please. And now already showing data of the peer-reviewed version of the preprint, in which we added this table. So NOVOS Core is composed by 12 ingredients, and in here you see 11 of them in the first column. And then on the next two columns, you see the preclinical effect in terms of lifespan assays. So this means that all the ingredients that we selected, and that's actually one of the criterion that they were selected based on, is that they extend lifespan, as you can see here, in different organisms. And finally, we were also able to map, based on mechanistic studies of those very same research papers that did the lifespan assays, we were able to get a hint of how they are able to extend lifespan, and we mapped them to the hallmarks of aging, the framework that I showed before. So here you can see 8 hallmarks of aging being targeted in preclinical models. So with that being said, I think we are fully covered in terms of prerequisites to understand the talk. So it's my pleasure to pass the word to Professor Satomi Miwa.
Hi, everybody. So now I'm going to talk about our recent mouse study using core, NOVOS Core, multi-ingredient nutraceutical, which we call it. The question we wanted to address is, does core have anti-aging effects relevant for us? So we wanted to find out the relevance for us. So the subject should be people like us, and how to test. Because it's anti-aging effect, we wanted to monitor how we age. And this is next, please. And how do we translate? So NOVOS Core, I don't know if you have seen it. This is a powder, orange flavored. And the instruction says it's put in a glass of water. And then you just drink it. But in mice, we do not know how to drink water or how much. We cannot regulate. So the way we give NOVOS compound, NOVOS Core to mice, is to mix with food and then mash up. And it's like a porridge, and they give it to mice. But this is rather appetizing, tasty. They love it, and they tend to eat a lot of it. And then they tend to become obese. Next, please. So this is a model used. It's people like us. Basically, we love soaked and with stodgy food, and then we don't do much exercise in a animal cage. So it's a bit like a couch potato and get a bit obese. So this is a subject, people like us in a mouse version. How to test anti-aging efficacy, which were established by our group, measures of phenotypic aging, so that's survival, fertility index, that is adapted to mouse version from a clinical fertility index, and the cognitive function, and cachexia, weight loss, which is also very, very relevant to human aging. Next, please. That's good, but we also wanted to know a positive control so that we can compare the experimental efficacy and robustness against the core effect, NOVOS Core effect. So for the positive control, we decided to use, next please, xenotherapy, which I'm going to just briefly overview because we have done many experiments using xenotherapy to delay aging or prevent age-related diseases. So for us, this is a really, really good positive control. Next, please. So what is xenotherapy? Basically, this means targeting senescent cells, xenotherapy. So senescent cells, probably you might have heard about senescent cells before, but normal cell, blue cells, can become senescent status in the red middle by telomere attrition or DNA damage or stress. Can be oxidative stress, oncogene, but high-fat diet or obesity can also be a metabolic stress to drive many cells to become senescent. And the senescent cells are different in looks and functionality. They tend to be larger, and the nucleus is deformed and larger. And functionally, they have mitochondrial dysfunction and pro-inflammatory cytokine production and reactive oxygen species production. And basically, they are very reactive, and that phenotype is collectively called senescent-associated secretory phenotype, SASP. And this, because of these SASP factors, senescent cells affect surrounding cells to become senescent. So senescent cells has bad effect about neighboring cells, and because of those SASP factors can travel across bloodstream, affect the distant organ, and that's called the bystander effect. So senescent cell can spread across the body. Next, please. Recently, a class of drugs called senolytics, senescent lysation, has been developed that targetedly kill senescent cells. And by effect, because we are eliminating senescent cells, we can prevent SASP spread. And this has been shown in many preclinical animal model, mice models, that can prevent many diseases or cure many already developed diseases, suggesting senescent cells are causative to many age-related diseases that include obesity-induced conditions like fatty liver syndrome. Next, please. As well as senolytic, which kills senescent cells, there are another class of drugs called xenomorphic or xenostatic that instead of killing senescent cells, that kind of mask senescent phenotype so that they don't produce SASP factors around. So they are still there, but they do not have a bystander effect or SASP. That has been shown to be effective against many age-related diseases too. Next, please. So we decided to use the senolytic treatment, the kill senescent cells, against NOVOS Core to see anti-aging effect in normally aging, bit obese mice. And here is a study design. And mice, we started already relatively old age, which is 20 months in mouse, which is about 60 to 70s in human. We gave senolytic in pulse, so that's 5 days of senolytic drugs. And at the same time, we started, it says nutraceutical, but it means NOVOS Core, mixed with food, and they are given every day, like NOVOS Core as taken by people every day. And this is lasted until 20 months to 30 months, which is about 85 plus years in human. Where senolytic treatment, we gave once in 20 months, and again 5 days at 23 months of the age. And we measured how they age in different rounds of assessments, around 1, 2, 3, 4, every 2-3 months. And the measures measured, as we mentioned, like clarity, cognitive function, and body weight, and those are aging characteristics. Next, please. So firstly, this is a snapshot of survival. And you see, control is blue, and green nutraceutical, which is NOVOS Core, green, and pink is senolytic. And the senolytic treatment was given here and here. And NOVOS Core has been given across all this study time scale. And don't know if I have to explain, but it's clearly both nutraceutical core and senolytic extended lifespan. Again, the control here, blue. Next one, please. This is a bit of a snapshot, and this is a bit of a catch. The reason I'm showing this is that blue one now is normal food control, which is not soggy soaked food, but normally fed. So they were not obese, normal kind of weight, typical mouse weight. And that's now in blue. And black is the current study control, which was fed with a soaked food diet. And again, nutraceutical, NOVOS Core, and senolytic, I forgot to mention, but senolytic mice also received the same diet as control, so it's a soft, and then they were obese. And as you can see, actually, nutraceutical senolytic mice, they did not live longer than normally fed control, but kind of normalized or prevented premature early death of control, which is obese. So what this data shows is that core and 2 times senolytic treatment normalized shortened lifespan due to obesity in these mice. So blue one is our historic record of our typical standard condition mice. So we are directly comparing diet and the treatment effects. Next, please. Yeah, so this is in numbers. So control, in this current study, was 703 days, median days, whereas NOVOS Core treated mice at 830 days in median. And the senolytic was also similar, not too much better than Core, 834. And this is I must say, this was surprising to us. We know senolytic has a very good health span extension benefit, but I was quite suspicious if nutraceutical can do something like that. And that was, for us, it was a surprise. Next one, please. And this is a body weight and age. Again, this blue line is control in current study, green NOVOS Core, pink senolytic. And there are a big error bars, but that's because it shows natural variation of individual animal, and that's typical. And as you can see, in average, control blue line start losing weight after 700 days, whereas the nutraceutical NOVOS Core and senolytic treated mice does not go down in body weight, which is a very, very important thing for all the animal and people as well. So the classic feature of people aging and nearing death is loss of body weight, body fat, and get very, very thin. The same thing happened to mice. And once mice started losing weight like that, it's telling us, oh, they are not very far from dying. Whereas treated mice did not show this until really late age, so they were fitter. Next one, please. Now come to frailty index. That shows very good overall frailty measures. We categorized 30 different classes of aging parameters and average it. And this has been shown very well correspond to healthy aging. And how do we see it? This is frailty index in y-axis, and the black line is control mice. And each dot represents each individual animal. And then this is a time course development of frailty index goes up with age. And the left, it shows senolytic treated mice. And then after 2 pulse of senolytic treatment, as you can see, pink line is a senolytic treated mice average. As you can see, frailty increase has been sharply dropped. So it's very slowed down the acceleration of frailty index with age. And right, green line shows mice treated with NOVOS Core. It doesn't make a big difference, slightly lower the increase in frailty index. So that was the data from round 2, 24 months, as you can see in the top figure. Next, please. So this is the next assessment timeline at 3 months later. For our a little bit of surprise in left, senolytic treated mice, we lost statistical significance. Now, look at the right hand, the NOVOS Core treated mice had increased the gap between control and the treated mice. Now, p-value is greater in NOVOS Core than senolytic. What happens? And next one, please. Next slide, please. So this is a further 3 months down the road. Left, senolytic, lost even more differences between control. So it's now very close to control. And p-value is getting worse. Whereas NOVOS Core, now, to our surprise again, the gap between control and the treated mice widened. And now p-value is reaching almost significance. Next one, please. So this one, we put the data all together. Top one is the time course changes is the senolytic treated mice. So the beginning left one is the largest difference between controls after shortly before senolytic second pulse. But the effect is getting lost with time. Whereas below is green one, NOVOS Core treated mice. So with the time, as you can see, the difference between controls and the NOVOS Core treated mice widen. And then after a long time, at 30 months of age, which is about 85 plus people age, it's almost significantly better. Better means slower rate of frailty index increase. So what it clearly shows is that 2 different treatment, both has extended lifespan effect and prevention of body weight loss with age, but also the way it causes the effect has distinctive patterns. Senolytic sharp change, but if we stop treating, the effect is lost. And then NOVOS Core, on the other hand, the effect is getting better and better. Next one, please. So final data I wanted to show you from mice is cognition. So cognition in mice is tested by Y-maze test. Mice are generally curious animals. They tend to love exploring new area. So normally mice explore new area, but if they forget which one they went, they keep going. So that's called spontaneous alteration we measure. And we tested mouse cognition in this round 1 and round 2 assessment, flanking senolytic treatment time. And here, round 1, so this data shows the difference between pre-treatment, which is about baseline assessment, which is 18 months. And here, round 1, there's no remarkable difference, but round 2 by 24 months, control mice lost cognitive functional ability, whereas NOVOS Core treated mice did not lose cognitive ability. Whereas senolytic compared with control mice, it was better functional cognitive functional capacity. we could not assess later on. It might have been different pattern. Maybe senolytic started losing cognitive function with time, whereas NOVOS Core treated mice might have improved better. We could not test just because we did not have enough animals left by later time from controls. So that was another surprise. Controls started dying much more than NOVOS Core or senolytic treated mice. So unfortunately, we did not have time, and we did not expect this much difference in survival between treated mice. The next one, please. This mouse study was quite explorative. We did not know what to expect from NOVOS Core treatment, so we did not plan to do detailed investigation of mechanism and any furthermore tissue level studies. However, we turned cell culture model to see potential mechanisms, how NOVOS Core made such a good anti-aging benefit. And because of the benefit pattern looked like our previous study, which used xenomorphic, xenostatic metformin, we wondered if NOVOS Core had xenostatic effect on cell culture level. So this busy slide I will explain now quickly. Right-hand side is senescent cells, and the left-hand, loss production, just to focus on loss production. And it measures reactive oxygen species production by cells. And the right hand is senescent cells, and left is non-senescent cell control. And we're looking at senescent cells. And because NOVOS Core, some of the compound was dissolvable in water, but whereas some others are only in ethanol, we prepared NOVOS Core ingredients in both water solubilized and ethanol solubilized version. And here, a water solubilized version, which is without stroke, in blue, that is a control, and the green one, water solubilized NOVOS Core, significantly lowered loss production by senescent cells. Whereas control cells did not do much. And ethanol treated cells, ethanol dissolved NOVOS Core did not show anything. However, what we think is that ethanol on its own is causing some problem, toxicity. What we can see, young cells, non-senescent cells in left side, looking at the blue control in water and the blue ethanol control without NOVOS Core, we can see just giving ethanol vehicle to young cells, non-senescent cells, increase loss production significantly. That shows toxic effect or side effect. So we think in senescent cells, the reason we did not see any effects of NOVOS Core might have been partially due to toxicity of ethanol as a vehicle. Similar thing was seen in IL-6 secretions. This is a cytokine, typical cytokine produced by our cells, fibroblasts. Quite strongly in water solubilized NOVOS Core, these cells produce much less IL-6, interleukin-6, as you can see here. Whereas young cell, non-senescent cell has already low production of cytokines, so we did not see much, which is not surprising. In ethanol solubilized NOVOS Core, again, we did not see any effect, but again, that might have been to do with the ethanol vehicle toxicity. So we did not conclude, we cannot conclude anything further in ethanol solubilized NOVOS Core treatment. But what we can say from here is that as long as the NOVOS Core has been dissolved in water, it has a capacity to decrease loss production and cytokine IL-6 secretion in cells in culture. We cannot say this is what's happening in vivo, in animal, but what we can see at the cellular level, this is what's happening after treating cells with the NOVOS Core. Next, please. So this is a summary from what I was just explained from mouse study. So the dosing, comparing senolytic and the NOVOS Core treatment. Dosing was different. Senolytic was 2 short pulses. NOVOS Core continuous and daily treatment. Time course effect of senolytic was immediate, but then fade if you stop treating animals with senolytic. Whereas NOVOS Core we have say gradual accrual of benefit, and survival both senolytic and NOVOS Core to similar extent increased against the control, so that everybody was fed with this obesity-inducing diet. Weight loss with age. I'm not going to say about weight gain by eating more food, but this is specifically for age-related weight loss that has a prevention effect in both senolytic and NOVOS Core. And frailty trajectory, and strongest effect after dosing was seen in senolytic, whereas NOVOS Core attenuated slope over time. And the cognitive function improved, at least what we can say was shortly after the treatment. But then we cannot say, we don't know. And NOVOS Core, cognitive function was maintained up to 24 months, and then we don't know. We could not test against the control. It might have improved, but we do not know for the moment. And mechanistic hint from a cell culture in vitro, senolytic is senolytic, but NOVOS Core may have a senostatic effect because we have seen senostatic signature in vitro. And I'm passing back to Diogo now.
All right. Thank you, Satomi. So to wrapping up, recapitulating a little bit of what Satomi mentioned, plus moving on to the translational potential. So just to recapitulate, NOVOS Core, again, it was designed to target all the marks of aging, is a nutraceutical. This is in contrast with the typical senolytic interventions, which tends to be pharmaceutical drugs or even experimental drugs. While one is using continuously, in this case, a nutraceutical, and the effect Satomi showed that tends to get stronger and stronger with time. The other one has an effect, a short time, is a very short-term intervention, but then it wears out and it needs another pulse, another intervention to keep accumulating this benefit. Next slide, please. So just to talk about this rescue model that we have for obese aged mice and why it might actually be of an increased translational importance compared with the normal experimental setup. So as we know, there is an obesity epidemic. So these mice better replicate humans, at least in developed countries. This obesity mice is not done by genetically manipulating it, but just by the more natural way of doing it. We just increased calorie intake. And we can see that the mice, besides being obese, they exhibited all the other phenotypical changes that are seen in obese patients, like, for example, an increased frailty, or in this case, the frailty index, which has several components that shows that basically the body is suffering a systemic process. Next slide, please. So just a little bit more on the frailty index. So this frailty index was built by taking consideration age-related phenotypes that are common between mice and humans, and of course, are relatively easy to measure in mice. And it includes very broad dimensions of aging, and in this case, frailty, from hair loss to grip strength loss and so on. But the key takeaway is that in this study, not only the xenomorphic and senolytic interventions, not only did they show increased lifespan, they show increased health span too. Next slide, please. So we already talked about the difference between senolytic and senostatic, and between the combination that of senolytic Satomi used, and also NOVOS Core as a senostatic, and focus a little bit more on the on core, the mode of action or the mechanism behind this effect. It seems to be that indeed there is less reactive oxygen species. There is a reduction of inflammation as measured by IL-6, and there seems to be senescent cells with smaller nuclei, and there is no lysis of old cells. So all this together seem to point that the effect is cynostatic. Next slide, please. I already talked about how the effects get larger and larger with time in terms of NOVOS Core, and this leads us to the clinical translational hypothesis that if obesity leads to the accumulation and acceleration of senescence across different body tissues, a cynostatic approach could improve resilience before the large weight changes and also after in an obese patient possibly could ameliorate some of the damage that is done long term. So it could be a plausible adjunct to GLP-1 therapy. And of course, this is just an hypothesis, so it requires clinical trials with functional endpoints and biomarkers. Next, please. And so we have these two levers, which is the combination of what we've been talking. They're not proven. This is based on the data that Satomi created. And basically, there is the primary lever, which we envision is, of course, the very efficacious GLP-1 therapies, then supported by an aging lever. And this can be a senolytic therapy or a cynostatic therapy like NOVOS Core. The downside of senolytic therapies is that they tend to be quite intense, and they need, and also the toxicity side of the equation is much, much larger than a nutraceutical. And so I would say that in terms of translational potential, maybe NOVOS Core would actually have a larger translational potential in this regard. Next, please. And finally, all science has its own limitations. So it's good to acknowledge that this was done in only one of the genders, male mice, and we don't know the sex differences. We didn't evaluate anything for humans in terms of clinical efficacy. And so although we generated new hypotheses that we do want to test and follow up, we cannot use it as a representation of health benefits in humans. As Tommy implied, in this case, the mice lived so long and the intervention groups that the control group didn't have enough sample size, so the statistical power decreased in more advanced time points. The in vitro work used fibroblast senescence assays, so we still need to check if the same mechanism is in play when it's in vivo, and it still needs to be independently replicated by another group to make sure the findings are very robust. Next, please. So as next steps, we would like to replicate the study. Again, following up with all the limitations that I talked about. So in females, maybe diverse age groups, and again, diet-induced obesity models. We could always measure more things to try to pinpoint even better the mechanistic part, and eventually also run a human study, specifically in GLP-1 populations or pre-obese, obese populations. Also to add up that this was the first webinar of our webinar series. The next webinar is actually going to be sharing the results of a human clinical trial in a different area, but stay tuned because they are quite exciting. And next, please. Finally, the key takeaways is that in overweight age mice, in this model of metabolic stress, NOVOS Core improves survival. The health span benefits accrue gradually, so they require long-term administration, and they seem to be caused by a cynostatic effect. We also think that this rescue framing is clinically relevant. especially to the obese populations. And finally, we shared our translational hypothesis that NOVOS Core or senolytic therapies could complement GLP-1 therapies by targeting these residual aging biology mechanisms. And of course, we do need human clinical trials to test it. And next, and with that, we move to the Q&A. So I guess I open the floor. And I don't know if we already have any questions in the chat because I haven't been followed, but so Tommy and I are available.
Hello, Satomi and Diogo. Thank you very much for this presentation. So I have a couple of questions. So the first one, with this data, can we assume something about preserved lean mass? In obesity care, I worry about sarcopenia obesity, especially with the GLP-1 associated weight loss. So have we have any evidence that supports muscle or strength or reduce fat progression?
Yes, that's a great question because we have seen prevention of body weight loss. That has been our question too. We did not plan to do any muscle level analysis, or there's an imaging way to quantify muscle mass and fat mass, which would have been very good to do it. But because our study was kind of explorative, and we did not expect to see such effect to be frank. So we cannot tell, but that would be very good because it's a very good advantage over weight loss drugs, which has been shown to reduce muscle mass. So yeah, great question. But next experiment, we'll definitely want to look at those things.
Thank you, Satomi. And Diogo, you were talking about real-world target patients and human clinical trial. So with this side, older adults with obesity and fertility risk will be a good option for the human clinical trial, or what do you think about this?
Yeah, I think, well, ideally, what we show that if you get obese and you get obese and you're about middle age, let's say 60, 70, so Tommy's work shows that if then you were to start these kind of interventions, whether a NOVOS Core or a senolytic intervention, you would not see all the fair effects of obesity, even if you don't have weight loss. Now, the idea would be if we do a weight loss, you probably can get rid of a lot of these effects because they are weight reduction dependent, but you might also have another complementary effect in things that weight loss, the part of the biology that explained that increasing inflammation, this chronic burden of senescent cells in the human body, this could possibly be addressed by this hypothesis. So basically, pre-obesity, obesity patients or patients that had a huge weight loss because it means that they were obese some time in their life before, they could probably benefit from these kind of interventions.
Thank you.
Hugo, ask away.
Okay. So, Diogo and Satomi, thank you very much for sharing this. I'm looking forward to download the paper. I have a few questions. So, what was the senolytic treatment that you used? And did you try to change the dosing of your core? So, did you try different doses? And do you see an effect on with the dosing? Yeah.
Hugo, that's a really good question. So we did a dose finding study, what we call dose finding study, preliminary before conducting this experiment. We did not have time and money to do actual effect of dosing on aging effect, but we measured toxicity or aversiveness from my stop eating. So if it's too much, normally, if it's an adverse effect or adverse feeling or something uncomfortable to animal, they will stop eating. we can quite easily spot by measuring body weight changes with different doses, which we did. So we have tried this dose we used, and half the dose and twice as much. And then we did not see any mice refuse to eat. So that's the reason, only reason we used. And another reason is that it's characterized by historically human to mouse conversion. So if you are giving this dose to human, it's 12.5 times to mice, because mice has a much higher metabolic rate. So this conversion factor has been quite often used for this kind of supplemental study. It's not always used for like a cancer drug conversion. It's normally given the same dose as a human, like mice. But for those kind of nutraceutical, those conversion typically is 12.5 times, which is we use. But we tested lower and higher dose for looking at how they respond.
And Satomi, what was the senolytic treatment? Maybe I missed it.
No, I didn't say because it's a bit complicated. This is a combination of very low dose of navitoclax and mitochondrial uncoupled above 15. So this is a study showing this combination allows to use much lower, 100 times lower navitoclax dose, because navitoclax has a toxicity known as thrombocytopenia. But reducing the dose makes it safer. But in order to maintain the effectiveness, we combine with mitochondrial uncoupled to give mitochondria stress. And then because the senescent cells mitochondria is already dysfunctional and they cannot cope with mitochondrial stress, the combination of those low dose navitoclax and mitochondrial uncoupled is very specific to senescent cells, and yet very, very effective. And that effect is even better than high dose navitoclax. And navitoclax and the dasatinib comparison shows that navitoclax is superior than quercetin, dasatinib quercetin mixture in our hands. So, yeah. So we believe this is a very good control.
So can I just it's a challenge for NOVOS more than maybe for Satomi as a researcher, but you are targeting obesity, like obese people, and you mentioned pre-obesity could also be targeted. Makes sense. So how about knowing that maybe 70% of us will develop some preclinical obesity at some point? You are not planning to try, like, let's say, healthy, even pre-obese schemes to see if you prevent actually even the appearance of obesity? Because obesity, the appearance is quite complex. It's more than calories in, calories out. So did you consider to try it before animals with 20 months and obese? No.
Yeah, it's a good question. I think Tommy can talk more about the study design, but in terms of
I can say it's a very good question. This study was aimed to see anti-aging efficacy originally, so we did not know. But having seen our results, definitely prevention is better than treating, of course, and that's a very good point. It might have, if NOVOS Core indeed target many aging mechanisms, that makes sense that it has a metabolic benefit. And then that might prevent, obesity to start from,
Yeah, I agree with Tommy that basically my hypothesis is that you probably would still gain weight, but you because you start even before the onset of obesity, you probably even have more amelioration of the obesity phenotypes that are not weight related.
Ines, looks like you've raised your hand. You have the floor.
Yes, hi. Well, as an outsider, I have a question. That is, what is exactly core? Is it a supplement? Is it a medical food or something else?
Thank you, Ines. So core is one of our products. Tommy showed that the form is in powder for the consumer, but in terms of actives, it is a complex of 12 natural ingredients. And so they were being selected because all natural, they've been used for a very long time. They don't have basically any non-side effects. The dosage are also smaller because they are synergistic to the formula in itself. And so it shows a much better toxicity profile than typical senolytic interventions that are pharmaceutical grade drugs or experimental drugs.
Okay, and another question that I have is about basically sleep, fatigue, and the quality of life. I think these are things that most patients, they care about. So, any human signal or planned outcomes?
Yeah, so we're going to have for the next episode on the series, we're going to share human clinical data. But Tommy shared the frailty index, which is the best and the most common proxy used for healthy years in the life of humans, because those are characteristics that both mice and humans have in common. For example, hair loss with age, muscle weakness with age, and so on. But Satomi, can you elaborate more? This is your expertise.
Yeah, as for frailty index, it's a very good predictor for how long they still live. It's been shown in human and mice that frailty index predict 5-year survival very well, as long as they're showing functional capacity of age. So that's why it's called the health span indicator quite often. Yeah.
Okay, thank you.
I have a brief question. So given the multi-ingredient nutraceutical that is NOVOS Core, how do we know which components are driving the signal?
Well, it's a very good question. So we also wondered, is one compound just doing it, or are there additive or rather synergistic? And if it's synergistic, that's most exciting. Well, but a lot of the potential toxins also, there's some small dose stimulate natural resistance mechanism, stress resistance to make body stronger. And it's also possibility given many natural compounds does this something called the hormetic effect. And those are two things that we are interested, whether are they acting because it's a small dose of each ingredient, are they have some stress response inducing capacity, or are they synergistic function, or just each one acting individually? And that's very interesting about mechanistic side of next step, I guess.
And Satomi, didn't you also assay all the individual compounds?
Yes, sorry. Yes, we actually tested individual compound to see whether they have senolytic function in cell culture, but we do not see any senolytic. So we know none of them on its own act as a senolytic. And we also tested in vitro or the NOVOS Core mixture on senolytic function, but we did not see any capacity for NOVOS Core to kill senescent cells specifically. So something else, yes. Thank you, Diogo. That's a good point.
Should we, if there are no more questions, should then we finish this episode?
I was about to say, if we have no more questions from the audience, Dr. Diogo, Dr. Satomi, just want to thank you both so much for your time and for being available to discuss the exciting medical learnings that are coming from the NOVOS team. We'll be sure to follow up accordingly with not only the recording, but also key takeaways from this time. And again, thank you so much for the availability to speak to what we are doing here at NOVOS and to these very exciting mouse studies, and also give a peek under the tent as to what is to come as we continue to provide these webinars to the medical community.
Thank you for opportunity. Bye.
Thank you, Satomi.
Thank you.
all the audience.